Dear students, I hope you all will be fine. I'm here, Muhammad Jamal Khan, lecturer in botany, University of Education, Lahore. Today, we will discuss a topic from one of the core subjects of botany, that is plant systematic anatomy and development. The course code of this subject is BOTN 1112. So this is lecture number 20, that is related to the section of plant anatomy. In this lecture, we discuss the concept, structure and function of various tissues, particularly parenchyma. Before we go into our lecture, we should know about some specific terms and precisely the background of this lecture. So, first of all, one know about the phytotomy. Simply, we define it as the branch of biology that concern or that deal with the internal structure of plants and their parts. So, phytotomy is also referred as plant anatomy. Here we have the level of organization. Before we study the parenchyma, one know about this level of organization. As the smallest unit of life is cell, that is made up of atoms, and atom combines to molecules, and molecules combine to form cells. After this, different cells that perform specific function combine to form tissues, and then different tissues combine to form organs, and after this organ combine to form organ system, and in the end we have a highly complex organization of the organism by the combination of different organ system. So here our focus is tissue. So what is tissue? Histology is the branch of science that deals with the study of tissue. And the tissue are in which the cells of plants are grouped in together with characteristics function such as photosynthesis, transport, and such group of plant cells combine to form tissue that perform a specific function and that are similar in structure. So here we have types of tissue. We have two types of tissue. One is plant tissue, the second one is animal tissue, but as far as concerned with this subject, we have a focus on plant tissue. Plant tissue. So the plant tissue is further classified into two types. One is meristematic tissue, the other one is permanent tissue. Meristematic tissues are those tissues that actively dividing itself. It further classified into three forms, that is apical meristem, intercalary meristem, and lateral meristem. In our later lectures, we discuss all these meristematic tissues in detail. But today, our focus is on permanent tissues. So, permanent tissues are the tissues that are derived from meristematic tissues and are composed of cells which have lost the ability to divide. These permanent tissues is further classified into two types of tissue. One is simple permanent tissue and the other one is complex permanent tissue. In this lecture, we discuss only simple permanent tissue. So a simple permanent tissue is that which is composed of same cells. Here we have three types of simple permanent tissue. One is parenchyma, the second one is collenchyma, and the third one is sclerenchyma. But this lecture is only about parenchyma. So the permanent tissues are those tissues that perform a very particular and definite function. In addition to these, these tissues have lost the power of division. And their cells are of definite form and size. Here we use term permanent tissue as they have the permanent shape, size and function. The permanent tissues are derived from meristem cells. So the process by which cells formed by meristematic tissues become permanent tissue is called as differentiation. These permanent tissues may be living or dead, thin-walled or thick-walled. So the permanent tissue is further classified into three types of permanent tissues. One is simple permanent tissue, the second one is complex permanent tissue, and the third one is the secretory permanent tissue. But here we have the simple permanent tissue. Very precise and brief definition of simple permanent tissue is that the simple tissues that are composed of one type of cell which have homogeneous or uniform structure and perform a particular function. So simple permanent tissue are of three types. One is parenchyma, the second one is collenchyma, and the third one is sclerenchyma. But in this lecture, our focus is on parenchyma. Parenchyma is a living simple tissue. Parenchyma is a cell, and you can say that it is a tissue type in which the cells have only thin primary wall, and these cells are unspecialized, lack the characteristics wall of collenchyma and the secondary walls of sclerenchyma. Cell have live nucleate protoplasm concerned with the various physiological activities in plants, and these cells are meristematic or permanent or simple homogeneous, composed of one type of cell only. Fundamental or ground tissue upon which other simple and conducting tissue appear to be embedded. These living cell contains plenty of water. They are thin walled cells, and These parenchyma cells consist of or composed of cellulose, pectins, and hemicellulose, and there is no lignin deposition in it. All cells are same, so cells are relatively undifferentiated, and it occupies in the ground tissue majorly. As you know, the parenchyma generally occur in the cortex and the pith of stem and roots, a mesophyll of leaves, a fleshy part of succulent fruits, and the endosperm of seeds. Epidermis, endodermis, pericycle, or other example beside these parenchyma cells are present in vascular tissue also, like xylem and phloem. So parenchyma of pith, cortex, mesophyll of leaves, and fluoroplasts originate from the ground meristem. And the parenchyma of primary and secondary vascular tissue is formed from the procambium and vascular cambium, respectively. And the parenchyma, such as phylloderm, arises from the phyllogen during secondary growth. So, as far as concerned with the distribution, a large portion of all plants organs are occupied by parenchyma, as I already told you. So as concerned with the origin, as I already told you that the parenchyma cells present in the primary plant body such as pith and cortex that originates from the ground meristem, while the procambium and the cambium give rise to xylem and phloem parenchyma of the primary and the secondary conducting tissue respectively. In leaves, protoderm and the ground meristem give rise to parenchymatous epidermis and mesophyll respectively. These parenchyma cells exhibit majorly a spherical shape, but in some cases it may be a polygonal or maybe a polyhedral cell. So here we have the different types of parenchyma. Parenchyma basically classify on the basis of two criteria. One is the function and the second one is the shape. When we talk about the function, we classify parenchyma into 7 types. One is chlorenchyma, the second one is parenchyma, then we have a prosenchyma, vascular parenchyma, medullary parenchyma, conjunctive parenchyma, and armed parenchyma. We discuss all these 7 types based on function of parenchyma very precisely one by one. So on the base of function, our first type is chlorenchyma. These are the parenchyma that possess chloroplast. And due to the presence of chloroplast, it performs the function of photosynthesis. These cells are majorly present in mesophyll shell tissues, and some of the chlorenchyma is also present in the outer cortex in young stem. These parenchyma cells are loosely arranged. Due to this, it possesses intercellular spaces. The second type of parenchyma on the base of function is parenchyma. These parenchyma cells possess a plenty of intercellular spaces, and these intercellular spaces appear as large air cavities. These parenchyma or parenchyma cells can store air, and these parenchyma or parenchyma cells are present in aquatic plants. So it helps the aquatic plant in floating, and the floating of such plants is occur by the phenomena of buoyancy. Respiratory gases are stored in parenchyma that can be utilized by the plants when required. Next type of parenchyma on the base of function is medullary parenchyma. These parenchyma are present in medullary ray of primary vascular tissues. These are elongated cells with very thin walled cells. Allow radial condition of water and minerals. These medullary parenchyma cells also store starch grain. The next we have conjunctive parenchyma. So the conjunctive parenchyma basically present between xylem and phloem in vascular bundles of fruit. So those parenchyma that are present between xylem and phloem in vascular bundle are referred as conjunctive tissue. These conjunctive tissue is involved in the formation of cambium in dicot stem. So simply, the conjunctive parenchyma play a role in the formation of conjunctive tissue. These parenchyma actually lignified parenchymatous ground tissue in which the vascular bundles are embedded in certain dicotyledons, and in monocots as the secondary growth is very limited or restricted, so these conjunctive tissues play a role in the secondary thickening of, in monocots and become sclerenchymatous when mature. The last type on the base of function of parenchyma is armed parenchyma. Armed parenchyma basically is a parenchyma which is found in the epidermis of leaves in some gymnosperm. These cells have many spiny projections within the cell, and the main purpose of this armed parenchyma, it is defensive in function. And here we have the example of pinus leaf, in which the mesophyll cell are parenchymatous cells that is present in pinus leaf as armed parenchyma. So on the base of their shape in cross section, we classify the parenchyma into two types. One is angular parenchyma and the second one is circular parenchyma. So the angular parenchyma, these cells are angular and polygonal in their shape. These cells are tightly or compactly arranged itself, so there are no intercellular spaces in it. The second, we have the circular parenchyma. These cells are circular in shape and they are loosely arranged itself. Due to this, it contains intercellular spaces. So here we have the image of angular parenchyma and circular parenchyma that indicates its shape. So now here we have the function. Here we have the function of parenchyma. Basically, the parenchyma is the fundamental ground system, tissue system, is based on parenchyma, or parenchyma form the fundamental ground tissue system, as you know, like cortex, pith, stem and root, or in stem and roots, and mesophils of leaf, endosperms, epidermis, endodermis, parenchyma. So all these ground tissues are the result of parenchyma. Next, we have the function of parenchyma is that it also acts as a storage cell. Most of the plant parts, such as seed, fruit, and tubers, are composed of parenchyma, and these parenchyma cells usually store starches, proteins, oils. Succulent plants such as cacti are composed of large part of parenchyma cells that store water. The parenchyma cells that contain chloroplast are photosynthetic, or they are also called as chlorenchyma cells. So these photosynthetic parenchyma or chlorenchyma cells constitute the ground system, like mesophyll of leaves, and they are further divided into two types. One is palisade and the mesophyll and the spongy parenchyma. Sorry. So here we have two parenchyma, palisade and the spongy parenchyma. The palisade parenchyma consists of closely packed columnar cell located beneath the upper epidermis, and the palisade parenchyma cells are rich in chloroplast. These are primary site of photosynthesis in the leaf, and so such parenchyma cells are called as chlorenchyma. Below this palisade parenchyma are irregularly shaped, widely spaced spongy parenchyma cells are present. So the spongy parenchyma cells are also photosynthetic, and the large spaces between these cells allow diffusion of carbon dioxide. And the parenchyma cells that have intercellular spaces, they are referred as arenchyma. In woody parenchyma is secondary xylem. The cells are thick-walled, and the parenchyma of aquatic plant have large intercellular spaces and is called arenchyma. So due to the presence of parenchyma, aquatic plant float itself on the surface of water through the phenomena of buoyancy. In some cases, these parenchyma also store some ergastic substances, and ergastic substances are those substances that are the byproduct of that are non-living byproducts, as intracellular deposits of starch or fat. So ergastic substances include crystals, oil drops, gums, tannins, resins, and other compounds. And some of these substances help the organism function and keep up cell structure, or are just stored food materials such as oil and fat. During the life of a cell, they can appear and disappear. Parenchyma cells also have a role in the formation of tyloses. As you know, tyloses basically are the outgrowths on parenchyma cells of xylem vessels of secondary heartwood. When the plant is stressed by drought or infection, tyloses will fall from the sides of the cell and dam up the vascular tissue to prevent further damage to the plant. Tyloses are formed when the protective layer surrounding the protoplast of the parenchyma cell proliferates into the vessel lumen following degradation and rupture of the vessel parenchyma pit membrane. So the role of the protective layer in the formation of sclerosed parenchyma. So this process is known as tylosis, and it is mostly occur in woody plants. So we can also say that it is the bladder-like distension of parenchyma cells into the lumen. As you know, all these meristematic cells are parenchymatous cells. So cambium is also a meristematic cell, and the parenchyma of cambium divide to form secondary tissue or secondary meristem. So parenchyma easily differentiated into secondary meristem.